Thanks for the invitation to be here and for these amazing introductions, Britt. It's been a pleasure throughout the day. Also for the very interesting program. I've been enjoying very much the multiple points of view that we've been hearing about today, and hopefully I will carve out just the other quartile that we haven't covered yet. So I wanted to talk about what we gain by building neural networks in real physical matter, or what we gain, or what we learn from that. So hopefully you'll recognize this is Alexa, and our current sort of best neural networks, Alexa, Siri, at the moment don't move around, right? And where we really would like to go has been imagined for us in movies like Ex Machina. Ava is a fully embodied robot. While I was making this slide, it occurred to me that all of our AIs are female, and I started to remember that 50 years ago we learned not to make them men because they will not open the pod door. And so we are built inside a body, and I think that this has obviously many implications for our nervous systems. Ultimately, we'd like to understand how our brain interacts with our body, provides it with a way of experiencing the world that's unique to being built out of biological material, and that's kind of where I'd like to go today. Before I do that, I want to just touch on a system that I will come back to later, and I think is a beautiful example of what we heard about earlier this morning, is that when we're thinking about brains, that's only actually a small fraction of the kinds of creatures that there are out there that do intelligent behavior. So this is a hydra. It's a cnidarian, and as you can see, or at least one might be able to interpret, it's doing kind of purposeful movement, right? So it does these kind of amazing somersaulting movements where it attaches its tentacles, it touches the ground, and then when it's ready, it flips its peduncle off the ground and attaches it somewhere else in order to locomote through space. Now, Hydra is an animal that has no brain. It has neurons that are connected together through a nerve net. And now, given all the cool new tools, it's a system that we're able to study in just exquisite detail. So what you're looking at here is Hydra in the hands of Rafa Justa's lab, where they have endowed the Hydra with calcium signaling in the neurons, in all of the neurons. And so now you're able to watch every neuron in the body. I mean, quite advantageously, Hydra is also transparent, and so you can see all those neurons directly with just relatively simple imaging. And so now we can watch the entire, I would call it a neural network, but people actually call it a nerve net because it's a much simpler construction than a typical neural net. These are spiking neurons. They're all connected together. They're connected together both by synapses and by gap junctions, which is something I'll hopefully at the end come back and talk a bit more about. Anyway, that is an animal in which we potentially can really understand the whole thing, right? We've got every neuron potentially mapped out. We can simulate all of these neurons. We can simulate their connections. And then we have to deal with the fact that these neurons are embodied in a creature whose own body provides presumably quite a bit of biomechanical feedback. So ultimately, one would like to solve the full neuromechanical problem of how this animal moves around. In any case, when we think about real intelligences, we realize that they are built from very different components than what we would build an electronic circuit from. So we heard mention of Ramon y Cajal earlier today, a Spanish anatomist who made these exquisitely beautiful drawings from microscopy of brains. And what you will see when you look at them is just the incredible sort of diversity of neurons that we have in real brains. Diversity both in terms of their geometries and, as we're learning over time, also diversity in terms of the ion channel composition that makes up each of those neurons, endowing them with a huge range of different properties. Now, when we go to build neural networks, what do? We build something like this, right? So this perceptron, back from the olden days, is definitely inspired by neuroscience. And in fact, as I think we've become increasingly aware, the field of artificial neural networks and of neuroscience have really developed side by side with a lot of back and forth conversation along the way. So neuron like this, a node in an artificial neural network, consists of these branches, which are kind of inspired by dendrites that take in inputs inputs via weights, which we can think about as being the synaptic connections. Those are all summed together here at the soma, here through this summation in this network function. And like neurons, we simulate that as being an excitable system so that there's some kind of threshold or activation function upon which an output is generated. Right, so all of that sort of has the spirit of a real neuron and sets up sort of the principles of how neural networks are constructed. So individual nodes sum their inputs and produce an output. These elements are connected together with weights that can be altered. And training a network or learning involves changing the strength of those connections. Right, so when we think about those connection strengths, that we've just heard a beautiful 1 hour talk about that. You know, if one can characterize a network completely through these connections, these connection weights, I guess what I'd like to argue today is that there's a lot more to think about, right, when we think about a real biological network in which the connection strengths are only a very small part of the problem. All right, so I just wanted to give you kind of a crash course. I know it's a bit supererogatory with Chris here in sort of where we're at with building neural networks, what's kind of the state of the art. And as everyone knows, these have evolved tremendously really over the past 5 to 10 years and have powers that certainly when I came into neuroscience were beyond anything we expected. Right, so going back many years, the basic form of networks were these sort of feed-forward neural networks, have some input coming in, gets mixed up in a hidden layer, generates an output layer. One can train the connections between these layers to produce pretty much any smooth function of your input. Those had limitations and weren't able to solve the kinds of complex tasks that we do in vision. In the last few years, people have gone back to this idea of being able to build a feed-forward network, but now endowing it with a lot more computational complexity, I would say, or not really computational complexity, just complexity, a lot more weights in which the computation is being done. Of course, these have been around for a long time, and we should really give credit to Canada for that. A lot of the original work on deep neural networks was funded through a Canadian research enterprise, and a lot of the members who are still involved in pushing these networks forward are still involved in that original group of people. All right, so what do these do? So you take an input. That input is filtered through a range of different sort of feature vectors passing through this convolution. Those outputs are passed up. They're convolved again with a different set of feature vectors. There's other operations like pooling and nonlinearities that happen from layer to layer, and eventually that's trained to be able to do classification. And these have reached a point in which they're performing at really remarkable levels of accuracy, and I'll show you an example of that a little bit later. As I said, the key players in a lot of this were from this original network: Jan LeCun, Yoshua Bengio, and Geoff Hinton, and you here in Canada. You've probably heard way more than you ever need to hear from me about all of this. These are forming the core of a lot of very interesting artificial intelligence research now at places like Facebook and Google. All right, so that's still very much in the spirit of feed-forward networks, and it's been realized in the last few years that there's a lot more one can do by building in something like memory. So again, going back to inspiration from real neural nets, what you saw from Olaf is that networks are generally not feed-forward, that they are recurrently connected. One neuron tends to be connected to another neuron, and there are connections back to that same neuron. And so a kind of new generation of networks are recurrent neural networks. So now in this hidden layer, these individual nodes all have interconnections between them. And what does that do? So yet another story to put another connectivity matrix on the screen, but it's not organized at least. So we have a connectivity matrix. What has that done for us in terms of the potential dynamical output of that neural net? And I thought Olaf might talk about this more, but so I'm just going to speak about it very, very briefly. But if you were to analyze what you expect to be the dynamical output of a network with connectivity like that, typical thing you would do is to compute its spectrum. And what would its spectrum tell you? All of these points represent the time constants of a given mode of the network. So the mode is able to generate many, many different kinds of dynamical outputs. They all come with an eigenvalue or a point on this two-dimensional plane, And that tells you the frequency and the decay time constant of that mode. And so what is particularly interesting in terms of trying to build memory is that if you look at modes that are sort of around this unit circle, those are modes that have infinitely long timescales. So where you sit relative to the unit circle tells you whether you're a mode that's going to decay away, so have a very short decay time. And if you're outside here, these are modes that grow in time, right? So they have and if you think about it as having exponential decay or growth, these are going to have exponential growth, whereas these inside have exponential decay. Here on the boundary, they're more or less stable, and they stay at a fixed amplitude. And so that tells you that now you have recurrence, you can generate outputs, dynamical outputs that have very long time constants. And that gives you the ability to take in information and to maintain it in the network for very long periods of time, which is exactly what one might want with memory. That in general is not the way that artificial neural networks have gone about incorporating memory. There's kind of a cooler, newer, more artificial way of doing that, these LSTMs, so long short-term memory. It's a particular design of a gate that allows networks to hold on to information for a very controllable amount of time. So those of you who don't know what that is, I kind of don't know what it is either, but I'm going to tell you about it. So input comes in here, it gets stored in this cell, and it will be held there unchanged. So there's another gate that tells you when that cell will take in an input. There's another gate that tells you how rapidly whatever is held there will decay away. And there's an output gate that tells you when that information will be released back to the network. And so this is like a little kind of holding pen for information, such that the network is able to kind of store that information, and when it doesn't want it anymore, it can forget it, and that information will be overwritten, right? So it's a kind of neat little sub-piece of the network that can now be incorporated to hang on to memory. So that has given networks a huge extra boost in their capabilities. So here's an example of what memory can do when you combine one of these deep convolutional neural nets with a language generating recurrent neural network. And you probably can't see that from the back, but now here's the caption for this figure. This is a woman is throwing a frisbee in a park. That's a completely machine-generated caption for that image. A dog is standing on a hardwood floor. A stop sign is on a road with a mountain in the background. A group of people shopping at an outdoor market. There are many vegetables in the fruit stand. Which is really kind of has moved us into a realm where we're happy to say that this is no longer just classifying dog, cat, right, which we're very excited about for a while, but is really moved a step beyond towards something that does start to look like what we thought artificial intelligence should look like. Here's another example where these types of networks are starting to do what we might think about as reasoning. Some of you might have seen that paper from DeepMind a couple of years ago, where now these networks, due to their memory capacities, are able to do what looks like kind of reasoning about, say, optimal routes to take on the London subway. Here's another use of recurrence that's having an enormous impact, probably on everyone's life now, is the recent work from Google on machine translation. So here again is a network where each of these pieces now is an LSTM, is one of these memory holding units. And so networks like this are able to take in sequences of information. This network, in fact, takes it and reads it in one direction, reads it in the other direction, operates on that, passes it through a bunch of filters, and encodes it and then decodes it. And I really can't explain this, but there's a great paper about it on the archive. And there you have a machine translation capability that's in fact able to translate from any language to any other language, as long as it was in the original training set, where it's not an independent model for every single pair of languages. There's sort of a universal model. We just need to change a parameter about which language you're using. So really very impressive, powerful capabilities. Let's go back to natural intelligence. So I'm going to really go way back down into sort of the weeds of natural systems and ask what it is that we have in natural systems that isn't at the moment being implemented in neural nets. And certainly not going to talk about things that have anything like the impressive capabilities of these type of architectures, but what are the dynamics that we're kind of missing in neural nets at the very lowest levels that may allow us in the future to put in extra capabilities. So how are biological neural networks different? one really important thing, and that's becoming increasingly more important as we all want to carry, right, an artificial intelligence in our pocket, is that they're incredibly energy efficient. And I'm not really going to say much more about that, because you can always talk to Chris about that, because that's kind of his life. They are highly adaptive. Individual nodes are diverse. So as I mentioned before, we have all these different cell types. Why does the brain have so many cell types? What does that capability provide? Neural nets our neural nets are able to learn from very few examples. That's not currently possible. If we think about the mechanisms of learning as being the types of synaptic plasticity that we know and love, those take many representations. So we know that neural nets, as learned artificially, take huge amounts of data to train. If we're thinking about long-term plasticity, LTP, as the biological mechanism that carries that out, that also takes many presentations for synaptic weights to be able to change. And that seems implausible as a mechanism. We also heard a bit about the role of neuromodulators. So node properties can be affected globally, right? So not everything is a kind of local transformation. We have the ability to change properties of nodes in a more global way. All right, so let's go into detail on a couple of those points. So what can neurons do? All right, so a few things that neurons are able to do. They can compute and combine multiple functions of their inputs. So unlike what we just saw from the perceptron that had a particular function, which is to sum inputs from its weights, neurons are able to do much more than that. I'll talk a bit about that. They're able to automatically adapt, something that all biological systems do very well, that artificial systems don't do well, is adapt, to take into account the environment and to continuously change the mapping from input to output based on what's happening right now. And they maintain memory of inputs over surprisingly long time scales. So that takes us to kind of the goal of the next piece, which is how do you even characterize neuronal computation? Right? We'd like to be able to think about neurons in a network in terms of an underlying computational goal, and the perceptron was one variant of that. More generally, how should we think about the computation that is being provided by such a complex physical system as a neuron? So as we saw before, perceptron is one idea. How can we improve upon that? And so we're going to use ideas that have been around for a long time in basic sensory physiology to replace the perceptron with maybe a slightly more powerful or slightly more useful model. And that model is this kind of cascade model. And you can see here that the perceptron is a variant of this kind of cascade model. But what I've done here is to draw, say, this is some complicated time-varying input, S of t. That time-varying input gets filtered through some feature. And now what we've changed compared to this picture, where you can think about these weights as essentially a feature over the space of inputs being given by these dendrites. Now our feature is some function over time. So we're looking for some variation in time, and the weights of this feature extract some function or some pattern from the incoming information stream. And like the activation function, we're going to put in there some nonlinear decision function that transforms the similarity of our input to a probability of firing. And that generates some output. All right, so what is kind of fun about doing this at the level of the single neuron is that in principle, we should be able to do this exactly, right? We know all the equations that govern the behavior of a single neuron. We can write down all of the biophysical properties of that neuron. And in principle, we should be able to relate all the components of a model like this to our coding model or our computational model. So I won't go through the math of how to do that, but it is in fact possible. And from that, we learn a few things. So we learn, for one, that neurons have the following properties. So they're multidimensional. Generally, when you think about what feature a neuron is extracting from a time-varying input, we often draw it as though it were one feature. In fact, we should properly think about it as being many features. The neuron is sensitive to different kinds of fluctuations and combines the strength of those different fluctuations in a nonlinear way. Right? So it's generally sort of wrong to think about it as just an integrator. There are generally many things going on that can influence the firing of the neuron. It's computationally robust. So there are many different underlying parameter choices, and those who follow the work of Yves Mader will be very familiar with this idea that there are many different ways you can set up the underlying biophysics in order to generate a particular computation. What's the space of particular computations that individual neurons can do? They can integrate, as we sort of put into our perceptron model a long time ago. They can also differentiate, and as we heard about this morning, they can resonate, right? So neurons can, they don't prefer resonances, but they are driven strongly, some of them, by inputs that have a particular frequency. So all of these types of computations are available at the single neuron level. They are subserved by many different choices of underlying parameters. But as you move around in neuronal parameter space, you can also dynamically change the nature of the underlying computation that a single neuron is able to produce. So let's talk a bit about this adaptive property. So neurons are adaptive. Here's a classical picture of adaptation, from 1929. The first time spikes were recorded from single neurons, this dynamics was observed, that if you turn on a stimulus, and this stimulus is pressure being applied to a cat's paw, while neurons that are pressure sensitive are being recorded, the firing rate goes up, and even though the stimulus is being held fixed, the firing rate goes down again. You drop the stimulus, you turn it on again, the firing rate will go up, and then again decay. That is one very classical form of adaptation, but over the last maybe decade or two, people have seen that there are sort of a little bit more sophisticated ways to think about adaptation, where systems adapt not just to the fact that a stimulus is being held fixed, but to the fact that the statistics of the environment are changing. So here's an example where I love this painting. This is a painting by Whistler of a girl standing in front of a window, I think in Venice. And when he painted that, he painted it as though he were a human being, which he was. So he could see the entire image at once, which is what you would be able to do in the same situation. If you were to take that picture with your iPhone, you can all imagine exactly what would happen, right? You would either get a very bright square in the corner and everything dark, or you would try and focus over here. You would never be able to capture the entire image at once, although our visual system does it effortlessly. So as one moves one's eyes around the room, if we capture a histogram of the brightness of the image, and we just look around this part of the room, we'll get a histogram that looks like that. If we look over here, we'll get a histogram that has a very different mean and variance. So what would we want to do if we were a neuron encoding that information, and we just thought we needed to encode this part of the room? Then we would build a curve like that, such that its dynamic range covered the range in which the variations in this image occur, so that we're using our firing rates to encode different levels of this image. Of course, now we look at the rest of the room. We're going to want a very different response curve, one that captures all of that the bright part of the room. And so if we'd used the wrong one, what would have happened? So if we now try and look at the dark part of the room using this red curve, you see all of these values in the blue histogram are down here at 0. And that's kind of what your phone is doing. You're setting it to one value or the other, and so you're completely unable to capture detail in the dark part of the room, or if you're using this curve, then you're completely saturated when you're trying to look at the bright part of the room. Neural systems, fortunately, don't do that. They have a lot more intelligent dynamics that allows them to continuously adapt their firing properties. And in doing that, if we look at it from the point of view of this kind of model, what they're doing is changing the properties of that decision function. And so we showed a couple of years ago that ability of a nervous system to continuously adapt to the statistics of the input is instantiated even at the level of single neurons. So this is data from a single cortical neuron where we're driving it with an input current that either is drawn from a distribution like this or a distribution like that. So with 4 different distributions, all of which have a very different variance, overall scale. And if you try to figure out what the input-output curve, given each of those stimuli is, you get these 4 different curves. Now, what is kind of beautiful is that if you take each of those curves and you divide the stimulus by the standard deviation of this distribution, in fact, it's always the same curve, but the neuron is able to kind of dynamically stretch the its input-output curve, or its dynamic range, to account for the range of stimuli that it's actually encoding at the time. And that has to be done purely by the biophysics of that single neuron, because we're injecting current directly into the neuron. We'd previously thought about this as being perhaps a sort of computational property that emerges from an entire network, but it's clear that it's a potential output of a single neuron. This is older work that shows that that is happening continuously and instantaneously. So this is now data from FlyH1, a motion-sensitive neuron in the fly visual system. We gave it a complicated time-varying noise signal where the variance of that signal is changing continuously. If we look at how the neuron is firing at different times throughout that presentation, we capture the input-output properties of the neuron in those different windows. It looks like it's changing continuously, and it is. And it's changing in a way that, again, looks like it's just simply taking that original curve and stretching the way in which it's encoding those stimuli to change the effective dynamic range. We were talking about this a bit over lunch. This is a property that is very widespread, not just across sensory systems, but even sort of cognitively. And it's harder to see that at the level of neural systems, but it's something that you should feel very familiar with. If you say you get a raise, right? You're very happy with your raise. Then you ask all your colleagues, how much of a raise did you get? And it turns out they all got 3 times as much as you did. You're suddenly not so happy, right? So in the value assignment system, we also expect to see at some level this kind of dynamics, whereby systems are continuously keeping track of kind of the range of values and assigning your happiness or your response to that in terms of what the local history or the ongoing statistics of those values are. So at the same time, neurons also have these slow time-varying changes. So we saw this, I showed you this picture before, and in fact, people often use the word habituation, and I put that up especially for you with the scare quotes, you know? So because it's not really habituation, and I think it's actually a wonderful example of your talk that choosing language that I think in your case was specifically intended to show that one was to be neutral a little bit to the meaning of it, right? You'd use a word that just described sort of what you see. In fact, habituation does carry an underlying valence, right? That the neuron is sort of stopping responding, and that's completely not in this case what's going on. And so we showed in other work that these long timescale properties of adaptation, in fact, are encoding the entire slow history of the stimulus. They're not just sort of pooping out or getting exhausted. They are continuously encoding through a very particular transformation the complex envelope of the input. So here what we did was to take a noise signal and modulate it with some kind of envelope. And so here we were changing it between 3 different values, and you see these adaptive dynamics. If one takes that original envelope of the noise and convolves it with a filter, which turns out to be fractional differentiation for those geeks who like this kind of thing, which is a kind of filter that is like differentiation, but is kind of a soft form of differentiation that has almost a power law-like nature to it, then you're able to reproduce exactly what those adaptive dynamics look like. And so that is just a pure linear transformation of the original data. No information is being lost in that transformation, in that adaptive transformation. And so what that shows us is that neurons are conveying information about these very fast time-varying variables in this kind of optimal way, and are also in their envelope retaining long timescale information through this slow, quite beautiful power law-like transformation. All right, so that's kind of complexities of dynamics at the single neuron level that we never put into a network. So what else can we see, or what other kinds of neuronal dynamics might be important to bear in mind or to realize that neural systems do? So one, neuromodulation. We know that neuromodulation can sort of switch properties of networks in interesting ways, and I'll show you one example of that. And also, we have the ability to hold memories, and because of these slow dynamics, one can even do that without synaptic change. So the example I want to talk about is, again, through dopamine. And dopamine has the role, it turns out, in the bird, of being able to change the bird's state from one behavioral state to another. So, this is an example where, say you're singing in the shower, you might sing in a somewhat less controlled way, or maybe more experimental way than you would if you were on stage. And the bird does exactly the same thing. So if you look at a bird singing alone, a male bird singing its song without the female there, it will sing in a more variable way than it does when the female is in the room. So here's Here's a spectrogram of the song. If we look at that syllable that's highlighted here and look at the fundamental frequency of that syllable, when the bird is singing alone, that distribution is quite broad, whereas when the bird is singing in the presence of a female, it's a much more constrained and more narrow distribution. And it turns out that if you play those two types of song to the female, she will preferentially she will look more often toward the loudspeaker that's playing the directed bird song. All right, so very quick crash course in bird neuroanatomy so that you know where we're talking about here. So quite a lot is known about the circuit for learning in the bird brain, which has made it just a fantastic system for theorists to try to understand where learning occurs. And usually a lot of the literature out there is about juvenile learning and how birds originally developed their song, and that's thought to be an example of reinforcement learning, and so there's a lot of interest about that. That's not exactly what we'll talk about here, although one of the fundamental properties of reinforcement learning is one that's relevant. And that is that every song is variable, right? And we've seen this behavioral difference in variability in the songs that the bird produces under these different behavioral contexts. Where is that variability coming from? So here, I've just drawn for you in red, these are the areas of the bird brain that generate the song, that drive the downstream motor, the syrinx, right, the motor neurons from the syrinx to generate the song. HVC is sort of thought to keep the timing of the song. RA transforms timing into output to motor neurons. These three areas are thought to be involved in learning and memory in the bird. In particular, you see that they're kind of outputting, so they get the timing information here from HVC, and they're outputting from an area called LMAN into the song. And that's where the entire output of that pathway comes from, and that's where variability gets injected into the song. So somewhere between these three areas, variability is generated, controlled, and output into the song to make the song either variable or not. Area X here is like mammalian basal ganglia, and it gets inputs from the VTA. And it is heavily innervated by dopaminergic inputs from VTA. So as in humans and mammals, VTA is thought to be responsible for kind of error signaling and for signaling of when you're doing a good job, right, of the value of your actions back to the basal ganglia to allow you to learn when to do well. So here, VTA is supplying dopamine. At the time that, in this terms of this contextual switch, we're not necessarily thinking about the bird doing active learning, but just being able to modulate its behavior from one social context to the other. What we see is that the output of that area X, is strongly modulated, strongly altered by context. So if we look at the output neurons here, a set of, they're called palatal output neurons of area X. In the presence of a female, they fire in this way, so you're just looking at raster plots of the firing of the palatal output cells. When it's alone, you see that the firing rates are higher and they're much more fluctuating. Right? You see these large pauses. And so we see already that at the level of area X, the output is distinct between the two social contexts, and that variability is larger when the bird is alone. It turns out that dopamine seems to be the signal that provides that context. So these inputs from VTA providing dopamine switch the bird's behavior from one mode to the other. So if we look again, so I showed you before histograms of that fundamental frequency of song when the bird is alone or when the bird's in the presence of a female. So here's when the bird is in the presence of the female, here's when the bird's alone. If we now take the bird when it is singing to the female and provide a dopamine antagonist, so we block the effect of dopamine, then that distribution goes back to very similar to what would happen if the bird didn't know the female was there. And so dopamine seems to have that role of providing the signal for modulation. So recently we've been looking at some neural data to try to understand why dopamine might have that effect. And what we found, and this is all work in Slice with David Bechtel's lab, is that these dopaminergic output cells receive a very strong excitatory input that hadn't been seen before, that is regularly timed, and very often happens in concert with an inhibitory input. And that what dopamine seems to do is to change the proportion of events that happen that are purely excitatory to ones that happen with an excitatory inhibitory pulse very tightly locked. right? So this is interesting, right? So with dopamine, the percentage of these linked events increases. So the way we can think about that, or at least sort of conceptualize it in a model, is that what dopamine seems to be doing is switching kind of this little mini microcircuit that's happening inside area X to a case where these output cells are receiving regular pulses from an excitatory, some excitatory cell within the area, to a case where it's getting both an excitatory and an inhibitory tightly coupled input at the same time. And these excitatory inputs, if you record from two palatal cells at the same time, you see that they're getting these excitatory inputs. Very often you see that they're happening at exactly the same time. So whatever that excitatory cell is, it's talking to many downstream neurons at the same time. And the same is true in this case. So dopamine is sort of having this effect of switching you from being a purely excitatory input to an excitatory inhibitory coupled input. We can use mathematical modeling to try to understand what role that might have. Because the output cells are firing periodically, and we're driving them with a periodic input, we can use a method called the phase response curve to try to understand the effect of that. And so what the phase response curve tells you is by how much the phase of the next firing of the neuron is going to change as a result of these incoming pulses. And so I won't do more than just sort of draw a picture for you of how that works. But these two different circumstances, where you're getting a purely excitatory input or an excitatory inhibitory input, end up having very different maps that take you from input to output. So in this case, over here, what you're looking at is how the phase of the firing of the neuron at the next time step depends on the current phase. And while it's being driven by this periodic background input. So let's choose some phase. We look up what the next phase should be from this map. And now we're going to take that phase and now look up. So now this is now phase n plus 1. We're now going to make it phase n by just reflecting on this line and seeing what the new value should be. Now it goes to there. We look up we move that on to be n plus 1. We look at what the next phase should be, and the next phase, and the next phase. And so what you see this map is doing as we propagate from one phase to the next is it's jumping all over the place, right? So we start at some phase, we're going to go from here to here to here to here. And the property of this map, the fact that it has this shape where it never crosses that line, means that every time we run this simulation, we're going to bounce all over the place. And if we start at a different place, we're going to go through a completely different trajectory of phases. That's very different from the case over here. So I'm going to go across. So here, let's choose a phase. And again, we'll do the same thing. We'll look up what the next phase should be, and the next phase, and the next phase. And we very rapidly, we end up at this red point, which is called a fixed point of this map. And we can start from a different phase. We'll go through a slightly different trajectory in the beginning, but we're going to end up at exactly that same phase. So these two maps have completely different properties. One is a chaotic map that's going to undergo a completely different trajectory every time, whereas this one has a fixed point where no matter where you start, you end up firing at the same phase and synchronously. Right, so just to show you that, just to draw out particular trajectories. So this is, as we move through iterations, what's the phase? Let's start two different initial conditions, one drawn in black, one drawn in red. You see the black one is bouncing around, through all these different phases. The red one's bouncing around. They take completely different trajectories. They never synchronize, and they're very variable, right? So both of them are, every single cycle, they generate a different phase. Whereas in this case, the two neurons, they might start differently, right, as they go through that initial part of the map, but they end up at the same phase and firing synchronously. So that gives us a mechanism for this action of dopamine, just by changing the effective weights of that inhibitory neuron, to generate completely different population dynamics in the output cells. So in one case, so we see both differences in intrinsic variability of the palatal output. So these are very different from cycle to cycle, and differences in the correlation between in the population, that every neuron here is going to be firing synchronously. So that tells us that the effect of dopamine in this case, it can interact with the physics of the palatal output cell to turn them from being regularly firing to being chaotic. And when they're chaotic, they desynchronize and they become variable. And so this shows us a way that we can have kind of chemical control of the dynamics of the network, which is something that you certainly wouldn't build into any artificial network there is now. But because we're working with physical systems, the opportunity to generate chaos and to use chaos creatively to cast your network into an asynchronous state is available. All right, so I've talked quite a lot now about individual nodes, how they sum inputs and produce outputs. So what we've said is that individual nodes are much more complex than just integrators. They can be differentiators, they can be resonators, they adapt, and they can change their properties through neuromodulation. So let's talk a bit now about how you train a network, which in all neural networks that are out there, what you do to train a network is you change the strength of the connections. So as I mentioned before, we've seen these very long timescale dynamics in single neurons. So looking at the timescale here, right? This neuron is changing its dynamics over timescales of many seconds. So this is just one example where single neuron properties can hold memory or convey information about its past over timescales that range out to many, many seconds. You can imagine through properties like bistability, right? If a neuron is able to be stable in two different modes, that allows you to flip it into a state where it will now hold memory over long periods of time. Changes in ion channel expression. You know, there's some long history of beautiful work about gene expression changes, changing complements of ion channels over timescales that are not that long, that then will change the intrinsic excitability of single neurons and thus change the way that node in the network functions. So we've been just playing around to explore the potential of networks like that don't change synapses, but just change their properties through hyperexcitability in this case. So if you, say, stimulate, you have a network where you've set it up with, in a very simple way like this, you stimulate some sequence through the network, through stimulus, right? So you take a stimulus, these might be neurons that represent something in the visual world. You have a sequence of events that happen in the visual world that drive some sequence through the network. But now these neurons retain memory of that event. Having been stimulated, they remain more excitable than they would be otherwise. So now you hit that first neuron again, because this guy is already partly excited just through noise, it will be more likely to fire again randomly. And so you'll trace again that output through the network. And so this can be used to do things that we see a lot in recordings of, say, spontaneous activity. So here's an example. You have, say, a neural network and you drive it. These little red arrows are stimulus. So you drive a sequence through the network. Now you drive the first neuron, and the rest of that sequence just propagates itself without any further input. And you can drive many different kinds of sequences through that network, sort of independent. I mean, as long as the networks have some kind of connectivity between them, this mechanism works just fine. You can then see these patterns reoccur in spontaneous activity. So again, driving that pattern through the network, and now you just watch the spontaneous activity. If you assume there's some kind of background noise in the network, you see that it will retrace segments of that sequence of activation. And so what that can allow a network to do is through this kind of short-term memory, right, that just comes about through sort of relatively brief changes in the excitability of the network, this could allow a neural network to kind of bridge out to those much longer timescales whereby synaptic changes can happen, because now just spontaneously you will revisit sequences over and over again. And so this might be a way to kind of bridge between the timescales of the occurrence of the actual event in the real world and the longer timescales that it needs to make change happen at the synaptic level. All right, so I just want to close up with just some speculative thoughts about the system that I showed you before. So I feel like we're sort of just on the threshold of really understanding the richness of the possible computational principles that are being embodied by real neural systems. And I think, all the talks we heard today sort of are pointing at that. So I showed you before this picture of Hydra, which is a great opportunity, as I said, to kind of dive into this in detail. So what I hope you'll see is that occasionally what happens is that you get these huge sort of coordinated firing events that seem to sort of spread throughout the whole network. very rapidly, right? So when it's contracting, it has this sort of huge sort of multi-neuronal firing event. So just watch it again. It's happened over, yeah. So here, it's almost, maybe a third of the neurons in the network are firing together. What we've been wondering about is the relationship between this sort of formation of coordinated ensembles in Hydra and the presence of gap junctions. In particular, one thing we know about Hydra is that it has gap junctional coupling among the neurons here in the peduncle. Now, gap junctions, for those who are not neuroscientists, mean that typically when we think about how neurons are connected together, we think about synapses. And synapses are chemical connections whereby when the presynaptic neuron fires, when if it's built up enough excitation to drive this sort of all or none spiking event, that will pass the message to the next neuron via a chemical signal that will travel across the synaptic cleft. Gap junctions are quite different. They instead continuously share information about the voltage in the neuron with the neighbors, with whoever they're connected to. And so rather than kind of waiting till they fire, then passing that message, they are always connected and always kind of a little bit losing, sharing information and also losing a little bit of their potential to their neighbors. So it's a very different way of thinking about neural activity, and one that we really don't ever build into our networks. So let's just, here's another example where we think that gap junctional coupling is responsible for the formation of ensembles or co-activation of neurons. And where this is in developing cortex. So if you take a slice of rodent brain early on in development, day P1, P2, what you see are these huge events, or at least large groups of neurons that fire synchronously together. And that only happens early in development. As the animal develops, those go away. What also happens is that neurons lose their gap junctional connectivity and start to be purely synaptically coupled. And so there's speculation, I don't know if it's been definitively proven, but that these large coordinated activity patterns are really due to this early level of gap junctional connectivity between neurons. No one really knows what that might be for, or what it might lead to, or why it's there. But it's very interesting, I think, that the sort of coordinated activity that we see in this very earliest nervous system is also present in the very early development of more sophisticated nervous systems. So I think there's some interesting thinking to be done about what might be the role of that. As our nervous systems develop, we lose gap junctional coupling among excitatory neurons, but it remains among inhibitory neurons. So that might be a way to distribute inhibition much more broadly. So, so what we see in Hydra is this rapid coordinated firing. Maybe that leads to this sort of formation of ensembles through gap junctional coupling. There may be a role, or maybe something interesting to be learned about this sort of interplay between sort of analog signaling between neurons as you get through gap junctions, and this more digital coupling that you get through chemical synapses. And it really does sort of take us back to the early day, again, going back to Ramon y Cajal, that early argument between Ramon y Cajal and Golgi, whereby Cajal advanced very strongly this idea that the single neuron was the computational unit. If it's true that gap junctional coupling is playing an important role, that sort of takes us back to the idea that maybe there's the syncytium, as Golgi called it, this sort of idea that neurons are not the fundamental computational unit, but maybe a slightly broader sort of dynamical system that's built of these multiple neurons. Anyway, so let's just summarize. So how are biological networks different? What have we said about that? So incredibly energy efficient, leads to us wanting to do neuromorphic engineering, which is a very important active area, in particular in the tech world right now. Individual neurons nodes are diverse. This allows networks to potentially pack in computational power. So if you want your network to carry out integration, differentiation, you actually have to build a whole network to do it, where neurons are able to do it just purely through their own internal dynamics. They're highly adaptive. That's presumably the substrate for the robust adaptive behavior that we see all the way up from the sensory to the cognitive level in animals. They can learn from new examples. I think there's thinking to be done about new modes of memory formation and how this sort of interplay between intrinsic properties and synaptic properties might sort of bridge the timescales that are needed to form new memories. And of course, this is definitely not a new idea. In the original Bliss and Lomo paper, where they introduced long-term depression, they were quite careful in that paper not to too strongly ascribe memory to long-term depression, but pointed out in the discussion of that paper that there are, of course, a lot of plasticity in intrinsic properties that one should keep in mind. And finally, nodal properties can be changed globally. You know, that really presumably can be simulated, but it's a very effective way that our nervous systems distribute messages over large parts of a network simultaneously, and in ways that have influences at sort of all levels of the network, changing the properties of individual nodes, changing synapses, and changing the dynamics of local microcircuitry. Right, yeah, so biological neural networks have dynamics that go really far beyond changes in connectome, and so it'll be, I think, interesting to see how many of, what of the richness that we know from biology we will see kind of propagating into artificial networks in the future. Okay, so that's my lab, and thank you for your attention. Yeah, how do you put it all together? No, it's a great question, and I certainly don't have the answer for it. Yeah, I thought Olive ended up with a very nice example of sort of taking what is known about the broad-scale connectivity and trying to marry that with, with adding in the dynamics at the single cell level, and trying to understand what will emerge from putting those two things together. I guess it's been the focus of my group to sort of try to articulate what are those low-level properties that we should understand, and trying to frame them in a mathematical language whereby it's not just a long series of differential equations, but we could say something a little more interpretive about them. I don't know what the right path, for anyone to go in sort of trying to put it all together. Certainly with respect to things like adaptive properties at the single neuron level, it would be worth trying to add that into a neural network and seeing what it does. I mean, a lot of the standard ways we had of thinking about neural networks and what we used them for until very recently was to classify, right? So there is no time in that problem. You just put in an image and you watch the output. I think we're moving into an era where we're doing a lot more interesting problems, right? We're looking at video, we're looking at language, and now time is becoming much more fundamental to that. And so I think it's a fun moment to step back and think about how we might enrich our idea about neural processing by incorporating time. Yeah, no, I think that quantum computation is a great example of the kinds of things that I've been trying to get at here, which is that physics imbues systems with properties that are a little bit surprising, right? That you don't necessarily build in to hardware. And I think neural networks are another example of that. Hopfield's original work showing that you couple together a neural network, you get these kind of emergent properties whereby the thing kind of you can self-organize to get states that now do error correction, all these kinds of things. So, I think that quantum computation is sort of another, it's another level of cool and hard, but it's in the same spirit as the way that we have been trying to think about using the physics of underlying systems to do computation. I think what will be an interesting challenge for quantum computation is sort of what are the right kinds of problems for which this architecture is going to give us a good solution. And I think we've been handling that and looking at that with neural networks now for a while, is what are the right problems that neural networks are the right way to solve, right? And I think we've made a lot of progress with that, and I assume that will follow as well with quantum computers. Right, in the sense that if you didn't have this mechanism, then you would drive the sequence in the first place, it would die out and you'd never see it again. Whereas here, what this would anticipate is that if the animal then shuts its eyes and goes to sleep, then these events will kind of just recur spontaneously multiple times. And every time they occur, they kind of bump up the probability of recurring again, right? So it's just a way of kind of reverberating that activity through the network over long periods of time that now bridge between the time it takes to give the stimulus and the time it would take to build up the synaptic connection changes, right? So that, I think it wasn't more profound than that, but that's the idea of sort of spanning out from presentation timescales to learning timescales via kind of low-level mechanisms for reverberation. And people certainly do see, Yang Dan and a group who I always forget his name, Houston, have been seeing reverberative effects in V4, V1, places like that. So certainly, hippocampus is obviously a place to think about it, but even in sensory cortices, these kinds of repetitions or reverberative spontaneous activities seem to happen. How we understand neural systems. Yeah, I know there are a lot of people out there for a while who've been hoping that like quantum mechanics is the answer to consciousness, for example, that there's some special thing that happens through quantum physics that might explain things we don't currently understand in the brain. I'm not a big believer in the idea that at least quantum mechanics is going to play a role. Although we shouldn't undervalue the importance of quantum mechanics for biology. For photosynthesis, and there's an amazing example where quantum mechanics and the physics of tunneling really produce a biological phenomena that's absolutely critical for life. I think in general in the nervous system, we're in a regime, at least I feel, that we're too warm. There's too much noise for anything truly weird and quantum to really happen. I mean, that doesn't say that there isn't physics of dynamical systems and statistical physics that we have not yet to discover. And I think that's probably true of sort of deeper understanding of how many interacting nonlinear things work together. I mean, I think there's certainly more to be done there. I'm not sure that it's going to come from quantum mechanics.